So good afternoon, everybody. Hope you're enjoying the meeting. So today I have the great privilege to introduce the 16th recipient of the AACR Distinguished Lectureship in Breast Cancer Research. Since 2008, the AACR has been distributing this special award to recognize outstanding science that has inspired or has the potential to inspire new perspectives on the etiology, diagnosis, treatment, or prevention of breast cancer. Since 2018, AFLAC Inc. has generously supported this special award, which contributes to provide an important platform to which recognizes and honors leaders in the breast cancer research field. Please join me in thanking AFLAC for their ongoing support. So this year's award recipient is Dr. Kornelia Polyak, who is currently Professor of Medicine at the Harvard Medical School and the Dana-Farber Cancer Institute in Boston, Massachusetts. Dr. Polyak is being recognized for her pioneering research involving the investigation of the role of the tumor microenvironment and intratumoral heterogeneity in breast cancer tumor evolution. She is revered for being one of the leading basic and translational scientists in breast cancer research for her consistent and significant scientific discoveries that have aided in the fundamental understanding of breast cancer tumorigenesis. In 2004, her groundbreaking publication, Dr. Polyak was the first to comprehensively profile and gene expression of distinct cell lineages of the normal breast, as well as in situ and invasive carcinoma, and this pivotal work resulted in the demonstration that the transition from in situ to invasive carcinoma is promoted by the loss of the normal myoepithelium. Furthermore, her studies beautifully uncovered the role of specific cytokines in the proliferation, migration, invasion of breast cancer cells. Dr. Polyak's comprehensive body of research has cemented her at the forefront of the intratumoral heterogeneity field. Her laboratory, for example, was the first to characterize CD24 positive luminal and CD44 positive stem cell-like breast cancer cells, highlighting the necessity of reconciling the cancer stem cell hypothesis with the clonal evolution model. She has since built upon this work with ever more comprehensive and sophisticated analyses of breast cancer tumor biology, using ecological and mathematical approaches and developing new techniques such as single-cell analysis. Her work has demonstrated the importance of polyclonality in metastasis and treatment resistance, identified novel treatment approaches such as bromodomain inhibition for TNBC, and latterly has substantially impacted our understanding of how the immune escape influences tumor growth. Throughout her illustrious career, Dr. Polyak has received multiple honors, including the AACR Award for Outstanding Achievement in 2007, the AACR Outstanding Investigator in Breast Cancer Research in 2012, and the NCI Rosalind Franklin Award in 2016. She was elected to the prestigious AACR Academy of Fellows in 2020, and in 2022 was elected to the National Academy of Sciences and the National Academy of Medicine. Dr. Polyak is the co-leader of the Dana-Farber Harvard Cancer Center Cell Biology Program, a member of the Dana-Farber Breast Cancer Program Executive Committee, and co-leader of their breast spore. And collectively, countless women have continued to receive better cancer care because of Dr. Polyak's unparalleled and groundbreaking research. And so it is my great honor to introduce Dr. Polyak as this year's recipient of the AACR Distinguished Lectureship in Breast Cancer Research.
Such a nice introduction. Thank you so much. I'd like to thank the AACR and the selection committee for selecting me as a recipient of this really honorous award. It was a really great introduction. I feel like I don't have to give my talk because it was all covered. But seriously, so do I use, oh, I use this one. Those were my disclosures. And then, you know, the awards are given to an individual, but I'm really, you know, accepting this award in the name of my lab. 25 years ago, we started studying breast cancer, and some of the lab members over the years are here in the audience. And we really had fun, and you know, all the talented and enthusiastic postdocs and fellows and all the other trainees is what makes this work fun to do every day. So this year was my 25th year anniversary. And of course, in addition to my lab, we have a really great breast oncology center. I think it's one of the best in the world. And just some pictures on this, I'd like to highlight particularly Eric Weiner, who of course started this breast oncology center 25 years ago, and then Judy Garbel, Marsbaum, Ian Kropp, and Partridge, and many others who had been really productive and fun collaborations over the years. And of course, we could not do everything on our own. We have many, many collaborators, and I actually generated this word art from all the collaborators in our publications. And I can highlight everyone, but a few I'd like to mention: Sarah Sukumar, Mina Bissell, Craig Allred on our Dcis studies, and Franziska Mikor, who we really got into the computational mathematical modeling of tumor regenesis. And of course, our funders over the years, we could not do any work without getting funding. And again, I just like to highlight that. So basically, what have we done in the last 25 years when I started my lab? So I did my postdoctoral training with Bert Vogelstein, and I was really intrigued by the Vogelgram of colon cancer. So I thought I will figure out, you know, the kind of Vogelgram of breast cancer. And in '97, there was a paper published by Mel Silverstein on Dcis that I particularly enjoyed. And I realized that this Dcis to invasive carcinoma transition, it's a really unsolved puzzle in breast cancer. And we started studying this, initially looking at the cancer cells. As everybody else, we thought there must be mutations or other alterations, and we did not find any. It was fairly disappointing. Dcis and invasive breast cancer pretty much looks identical in terms of the mutations and so on. Then we realized that a tumor is not just the cancer cells, but of course have many other cell types. We did the study with the purification of magnetic bead purification of major cell types and profiling, and then we noticed that there, of course, changes happening in all the cell types as well. And then the intratumor heterogeneity studies, me and others have seen that cancers are, you know, very heterogeneous. Even at one stage and one tumor, there are many different cancer cells with different properties, and these evolve over time. And we published a number of papers and particularly showed the functional relevance of heterogeneity by identifying non-cell autonomous drivers as one of the drivers of heterogeneity in tumors. And in the last decade, we became more interested in immunology in a way that, you know, we thought that the Dcis to invasive transition is really a key step of immune escape in breast cancer, because in Dcis, the cancer cells are still kind of within a duct, fairly shielded from an immune environment, but then when they become invasive, they have to survive and overcome the immune attack against this. And then as we studied this immunology, we also realized that cancer is actually much more than a tumor. We really have to study the individual, because even the host factors like pre-existing immunity could influence progression. And I will show you some highlights of some of our studies with focusing on more of the recent work in this presentation. So this was our first profiling of, again, like purifying the major cell types from normal Dcis and invasive tumors. And at that time, there was no single-cell RNA-seq, so we did SABCS. And then we found that the myoepithelial cells in particular had the most significantly differentially expressed genes between Dcis and normal, and many of those encoded secreted proteins. And then we did follow-up studies. Min Hu was leading many of those in the lab, highlighting that the myoepithelium actually seemed to be inhibiting invasion. And Dale Porter and Miller Allen were, again, key for this early work. Then looked at the heterogeneity of DCIS because we thought, heterogeneous tumors in an invasive cancer, we've seen that more likely to be resistant to treatment. So we also tested subclonal heterogeneity in DCIS invasive tumors. And again, I just highlight an example from this immunofish studies that was done by a very talented and very patient breast pathologist, Soyeon Park in the lab. And this was a nice example of how in DCIS you can have a subpopulation of amplified cells in a CD44 population, and that becomes a dominant clone in invasive tumors. So again, this was back in more than 10 years ago when we really thought that doing single-cell spatial profiling studies would be informative for this progression. And then again, we use these mathematical, ecological models to assess diversity, like the Shannon index of diversity with Francisca's lab. And then we showed that in DCIS, we see a very high degree of heterogeneity and independent of the tumor subtype. And then you see this plot where you can plot the species, individual cells, and you can tell that the populations change over time. The CD24 population here was very homogeneous. The 44 was more heterogeneous. And then in invasive, both populations became like this. So that, again, just highlights this heterogeneity in DCIS. But then coming back to our immune escape studies, so we thought that applying Bob Schreiber's model of steps of immune escape, in DCIS, when the DCIS is still intact, that could potentially be the elimination where the cancer cells that kind of get out would be able to eliminate by the immune cells. Then you may have an equilibrium in the DCIS IDC where you have both components in the tumors or microinvasive DCIS. And then, of course, invasive tumors are completely escaped the immune attack. And at that time, when we started these studies back in 2010-11, and then we thought to studying this immune escape would help us inform, how to treat more advanced tumors. And we got many rejections because everybody says that DCIS is not killing the patient, so why do we care? But our argument, and I will show you that if we understand how the DCIS escapes the immune attack, then we could actually design better therapies for more advanced stage patients. So here again, we started studying the normal DCIS invasive tumors. We did RNA-seq on purified T cells, and we found that the T cells from normal in DCIS and invasive tumors cluster very distinctly. We see many immune checkpoint inhibitors like CTLA-4 higher in the T cells from invasive tumors. In contrast, in DCIS, we saw a very active immune environment. We actually see these tertiary lymphoid structures very nicely. And T cells in DCIS have higher diversity compared to invasive tumors. And these studies were led by Carlos and Sung-Jin, postdocs in the lab at that time. Then we followed up and investigated what could be the reason of becoming more muted immune environment in invasive tumors. And one explanation is, again, the upregulation of immune checkpoint inhibitors. And CD274 is actually amplified in a subset of invasive tumors, as is shown in genomic data, almost never or very rarely in DCIS. And then we show this by immunofish also, that in DCIS, we almost never see amplification of CD274 and expression of PD-L1, but in about 30% of triple negative, we saw very high amplification and overexpression. So again, this could be one mechanism by which the invasive tumors escape the immune attack. Then we showed that the activated CD8 T cells actually very high in the DCIS in a triple negative HER2 positive subtype, and then they are almost absent in the invasive tumors. And this was looking at granzyme B positive, CD8 positive cells, or Ki67 positive T cells. And this was also true in patients who were diagnosed with pure DCIS and then recurred locally with invasive disease. In every one of them, this was a small cohort, but every one of them, we saw a significant decline in activated CD8 positive T cells. And we became also interested in studying, doing functional studies to show that this immune escape is actually relevant for the progression. So we started using this NMU-induced rat model of breast cancer, which was used back in the 60s or 50s, but forgotten. And the reason why we decided to use this, because the immune environment in particular and the location of the cells very highly resembles of the normal DCIS invasive tumors, like here when we stain for CD3, you see in the normal ducts, we very commonly see 3D positive T cells. In a DCIS-like lesions, they are excluded, just like in human DCIS, and then they intermingled in invasive. And then we also saw that for the tumors to grow, there is actually a significant drop in activated, again, Ki67 positive CD8 T cells, suggesting that this immune escape is very much resembles what we see in human. And we published a lot more on this that I don't have time to cover. So I just show you some more recent data, some of it unpublished and, or recently published. We went back and really, with this new technology, single cell profiling and also spatial transcriptomic, we revisited this normal DCIS invasive transition and did a parallel study in a human patients as well as in a rat model. The advantage, again, the rat model, that we can actually have very early lesions that we don't even have visible tumors analyzed, but I show you that the T cells are already active at that stage. So we did this comprehensive profiling. Triad Bue, a postdoc in the lab, has been leading these studies. And I just show you some highlights. So if we look at this UMAP plot of the single-cell RNA-seq data, again in a human and in a rat in parallel, and just coloring according to stage, you see that in every population we see normal DCIS invasive clusters, but in a T-cell population, it seems like the most distinct clustering according to histologic stage. And this was also true in a rat model where we have untreated normal mammary gland, NMU injected without visible tumors, and then in tumors as well, suggesting that maybe the immune populations, again, seems to be the most significantly different between tumor progression stages. So we reclustered these immune cells in more detail. And again, you see that the T cell is the major population, both in a human and in a rat. And we see very distinct stages of normal, mixed DCIS invasive, and then invasive only. And this is, again, seen as a rat as well. And then if we're reclustering T cells to see what are these different T cell populations, taking advantage of the really large amount of single cell data on T cells, if we split them up into CD4 and CD8, we see, interestingly, that in a CD4 population, there is a very large number of Tregs. In a human, 5 different subpopulations. In a rat, we actually see 7 different Treg populations. That seems to be the prominent T cell in the tumors. Then we use this Milo neighborhood analysis to basically identify neighborhoods of T cells according to, independent of the UMAP clustering, but more like defining stages of the tumor progression. And both in the patients and in a rat, we see that there is a normal neighborhood. There is a mixed neighborhood of the invasive DCIS, and then there is an invasive-only neighborhood of the T cells. And what are these differences? So if we plot here the DCIS invasive differences, T cells that are high in the DCIS are the CD8 cytotoxic T cells, as we've seen before, and also naive CD4s. In contrast, in the invasive tumors, there is a very large enrichment in Treg populations, and then we also see exhausted and stressed CD8 T cells. So this Treg population, particularly what we identified, we call it the cycling Treg, is particularly interesting because if we do this pseudotime trajectory, considering the starting point as the normal and then DCIS invasive, we got a very nice trajectory. The normal is very different. Then we see a bifurcation, and then you have the in DCIS direction and the invasive direction. And this cycling Treg seems to be in the middle, where we're basically deciding whether we want to, are these DCIS or invasive. So we think it's a critical cell type for the invasive progression. And we have a lot more data that I don't have time to show you, but we actually can deplete these cells and then in a rat and then show that the immunotherapy and tumor progression, we can prevent it, and that seems to correlate with progression. We stained for this cyclin Treg in Dcis samples and invasive recurrence, and again we see that this cyclin T invasive recurrence, and again we see that this cyclin Treg population seems to be more common. So this is Ki67, CD3 positive population seems to be more common in invasive recurrence. And what we're arguing and what we see in a rat model, that if you have a combined targeting of the Tregs, particularly this cyclin Treg, in combination with immune checkpoint inhibitors, you have a much more effective control of the tumor than either one alone. So basically, based on these studies, we think that the DCIS to invasive progression is really a key step in immune escape. And by better understanding this and better understanding which cell types like the cyclin Tregs and others could be critical, and we also see that there are some fibroblast and myeloid cells seem to be regulating the Tregs. If we could understand how those cross-talk to each other and establish this immune escape in invasive tumors, then we could, again, develop better treatment strategies for advanced stage disease. And then, seeing this big decline in activated immune cells in DCIS to invasive progression also raised the question, could it be that your pre-existing immunity somehow influences your probability of progression? And then could this be dependent on age and, could determine your age of diagnosis and the risk of progression? So we decided to do a study in collaboration with our great clinical colleagues, and Partridge, who's leading the Young and Strong cohort, and Nancy Lin, who's been leading the Embrace cohort in metastatic cancer. And again, we collaborate with Francisca's lab on a computational side, and this is June and Julie in my lab who led this study. And why are we interested in host immunity? So there is an increasing data suggesting that the host immunity is relevant for cancer. We know that growing up in a sterile environment it could increase the risk of childhood leukemia. Recent paper very nicely showed that removal of the thymus, in especially younger individuals, increases cancer risk, and also immune suppression. In breast cancer, prolonged antibiotic use have been linked to higher breast cancer risk. And then there are also papers showing that there is a T-cell cross-reactivity between bacterial and viral antigens and tumor-associated antigens. And the recent data from immunotherapy studies also shows that the blood TCR diversity could be predictive of response to immune checkpoint inhibitors. But the immune system also changes with age, and as we know, cancer is most commonly diagnosed in older individuals. And the one dramatic changes that happen, that we have a decrease in the naive T and B cells, and increase in exhausted T and B cells, and because of that, older individuals cannot mount a very effective immune response, as we have all seen in the COVID pandemic. And then there is also, more dysfunctional T cells in older individuals and increased transcriptomic heterogeneity with age. So that really suggests that, the combination of age and the T cell diversity together could potentially influence risk of progression. So the cohort that we designed, we really wanted to compare the two extremes of tumor progression. So we had DCIS and de novo stage 4 patients, because we also wanted to eliminate the treatment effect. And unfortunately, that was actually not difficult to have de novo stage 4 cases. And then we had young individuals, which we considered under 45, and then over 45. I'm not calling it old, but it's age. So this was a kind of a cutoff we used. And then we did, it's about 500 samples. We ran TCR-seq on the blood on everybody. And then we also ran RNA-seq, whole blood RNA-seq on a subset of cases to see how the TCR diversity could influence the whole blood transcriptome. And on a subset of DCIS, in the young cohort, we also did RNA-seq on the tumors themselves. what we found when we're looking at, and looks like the dots disappeared during transition from a Mac to PC, but you have to believe me, there are dots there. Basically, if we're looking at the age association of the TCR alpha and beta diversity with age, in both DCIS and stage 4 patients, we see a very significant decline of TCR diversity. There are many different indices you can use to assess TCR diversity. Here we use the CHA index. We can also look at richness or Shannon index, and you can also see the clonality. So clonality increases, meaning like you have more T cells of the same type. That increases with age, and then your diversity decreases with age, which was known as well. Then we also wanted to see if there is a difference according to tumor stage. So we looked at the CHA index of diversity in a young and old individual according to stage. And in a young woman, we saw that the stage 4 patient had significantly lower diversity. And this was also true when we combined the T cells. So you have a TCR sequence, but we can also assemble them into CD3 clusters because multiple sequences can encode the same amino acid in a genome. And then that one also, we saw that there's a significantly lower number of CD3 clusters per patient in the stage 4 patients, and this was significant in a younger cohort. But of course, we wanted to see if there is any association with clinical outcomes. So for this, we looked at DCIS patients who did not or had recurrence over the follow-up period. And then, in this case, again, looked at the CHA index of diversity. And then what we saw that in the older age cohort, the ones who had recurrence had significantly higher diversity. We didn't see this in a younger cohort, but one of the caveats is that in a younger cohort, many women had double mastectomy, which obviously we cannot see recurrence. But basically does suggest that maybe your diversity could determine your risk of recurrence, and we could, there are many reasons why it's actually could be higher diversity, higher recurrence that happy to discuss after the presentation how that could happen. And then the question was, do we have particular CD3 clusters that could potentially be associated with the risk of recurrence or the stage of the disease? And we saw that in an older woman, the stage 4 patient, if we're looking at the unique CD3 clusters, was significantly lower in the stage 4 patients. And then when we apply the kind of a predicted antigens for T cells, there is a database you can do predicted antigen recognition. Interestingly, we saw that in a younger cohort, DCIS patients, the ones with or without recurrence have significant differences in the predicted antigens that the T cells recognize. Of course, this database is very incomplete, but still it's kind of intriguing, potentially suggesting that there could be some antigen-specific T cells that could correlate with risk of recurrence in patients. Then coming back to the blood transcriptome data, we did RNA-seq about 100 cases, about 25 from each of the 4 groups, age and stage. And then when we're clustering this data, we did see some association of the clusters with age. But what was interesting that the clustering was more significant with TCR diversity based on CHA index, suggesting that if your TCR diversity is different, your whole blood transcriptome is different. And we see that the ones with a higher TCR diversity tend to have gene set enrichment in more T cell activation related pathways, suggesting that the TCR diversity could reflect the T cell activation or immune activation in the patients. And lastly, we wanted to see if there is any association between the TCR diversity in the blood and the tumor expression, and also particularly the CD8 T cells. So this is RNA-seq on a subset of cases in a young DCIS cohort, and we do see significant differences between a high and low diversity group. And the ones with a high blood diversity seems to be enriched in more T cell activation pathways. And then we also stained for CD8 T cells, and we see that if you have higher blood TCR diversity, then you have more CD8 T cells in the DCIS tumors, suggesting that higher diversity leads to more immune surveillance in the tumor as well, potentially explaining why this could correlate with progression. So lastly, I'd just like to summarize what I showed you today. Over 25 years, we've studied DCIS2 invasive progression. We see that there is an increasing immune suppression and changes happening in many cell types that could correlate with this. And then we also see high heterogeneity. And then I show you that the rat model could be used to do functional studies. And lastly, I'd like to highlight that we really need to look at the host beyond the tumor to look at markers of progression. And thanks again for this honor. And this is my lab, and this is the more recent people involved in this particular part of the work. Thank you.
Well, thank you very much, everybody. That concludes this session. If you have any questions, please feel free to come up to the front. Thank you, everybody.